Hello friends, subscribe to our channel GTC and press the bell icon for more updates.
Channel. So today in this video we are going to discuss the question portion with their brief explanation. So here we have a first question is that which of the following not causing enzyme inductions, and the options are: option A is phenytoin, option B is phenobarbital, option C is sulfonamide, and option D is alcohol. So the correct answer is sulfonamide, because sulfonamide is a enzyme inhibitor. So these phenytoin and phenobarbital alcohol are the enzyme inducers. So the correct answer is sulfonamide. So let's discuss in detail with an explanation part. That here we are having a table. In this we are having given inhibitors and the enzyme inducers. So let's have a look on this. That it's a trick to learn. Students just try to learn with this trick, so it make feel easy to get learn within minutes. So let's look on trick, that is don't join this group, it will make your spirit go down. So it is about 𝙂𝙋𝙼𝙏 group. We are talking about this group. Don't join these groups, and the group name is 𝙂𝙋𝙼𝙏 group. So let's discuss in detail that S stands for sodium valproate, I stands for isoniazid, C stands for cimetidine, K stands for ketoconazole, F stands for fluconazole, A stands for alcohol, C stands for chloramphenicol, E stands for erythromycin, S stands for sulfonamide. This question is asked in that sulfonamide is an enzyme inhibitor, and C stands for ciprofloxacin, and O stands for omeprazole, M stands for metronidazole, G stands for grapefruit. And where we are talking about the enzyme inducers, so we have to learn with this trick. CRAB, GPS induce me to madness, where C stands for carbamazepine, R stands for rifampicin, A stands for alcohol, P stands for phenytoin, G stands for griseofulvin, P stands for phenobarbital, and S stands for sulfonylureas. So it's all about the enzyme inhibitors and inducers. So you have to learn with this trick. It will make easy to attempt the question. So let's discuss next question, that the instrument used to measure a particle volume, and here we have options are: option A is Coulter counter, B is microscope, C is Hempel-Burette determinant, D is helium densitometer. So the correct answer is Coulter counter method is used to measure particle volume. So let's discuss each and specific use. That Coulter counter method is used to measure particle volume, where microscope is used to measure particle size, and Hempel-Burette is used to determine the heat of combustion of coils, and also indicated the possibility of using electrolysis of sodium chloride solution. And helium densitometer is used to measure the density. So the correct answer is the Coulter counter, which is used to measure the particle volume. So the next question is, when was the first train steamed off in India? So here we have options are: option A is 1848, option B is 1853, option C is 1875, option D is 1880. So the correct answer is that in 1853 the first train steamed off in India. So here we have next question is that which of the following is the largest railway junction in India? So here we have options are: option A is Delhi, option B is Bhatinda, option C is Mathura, and option D is Allahabad. So which is the largest junction, railway junction in India? And the correct answer is Mathura. Mathura is the largest railway junction in India. So next question is IUPAC naming of the ring commonly known as 𝘖𝙬𝙬𝙬 is. So here we have options are, option A is imidazolidine 4,5-dione, option B imidazolidine 2,4-dione, option C imidazoline 4,5-dione, option D imidazoline 2,4-dione, and the correct answer is imidazolidine 2,4-dione. So let's discuss with the help of a structure that imidazolidine ring is present in this structure. So the correct answer is imidazolidine 2,4-dione. So let's discuss this next question, that oxygenated blood returns from the lungs to the which part? And here we have options are A, left atrium, B, left ventricle, option C, right ventricle, option D is right atrium. Here we are talking about that oxygenated blood returns from the lungs to which part? So the correct answer is left atrium. So let's discuss this portion in detail. I hope that you will understand easily from this explanation that pulmonary circulation. So here we just talk about the circulation. Pulmonary circulation is the portion of the circulatory system which carry deoxygenated blood away from the right ventricle of the heart. Students, just try to focus that that deoxygenated blood away from the right ventricle of the heart to the lungs and return oxygenated blood to the left atrium and ventricle. That oxygenated blood returns in the left atrium and ventricle in the heart. So the term pulmonary circulation is readily paired and contrasted with the systemic circulation. Hence the vessels of the pulmonary circulation are the pulmonary arteries and the pulmonary veins. So from this that we can understand that deoxygenated blood level leaves the heart, goes to the lungs and then re-enters the heart. So how it is possible that deoxygenated blood leaves through the right ventricle? Just try to learn that deoxygenated blood leaves through the right ventricle through the pulmonary artery. It's many a time the question may also arises that from which artery the deoxygenated blood leaves through the right ventricle. So the correct answer is pulmonary artery. From the, so next proceeding, that from the right atrium the blood is pumped through the tricuspid valve or right atrioventricle valve into the right ventricle, and then blood is then pumped from the right ventricle through the pulmonary valve and into the main pulmonary artery. And then just we will discuss an explanation with the help of the diagram. So here we have a diagram that it's a right atrium and it's a right ventricle, and here we have a left atrium and left ventricle. So how it is possible that, let's discuss that it is having that the blue color is denoting deoxygenated blood. Okay, here we are, and the red color is having denoted oxygenated blood. Let's discuss in detail, students, that the right atrium having deoxygenated blood and it goes to the right ventricle via tricuspid valve or atrioventricle valve. Then when it is in right ventricle, then it goes through the pulmonary artery to the other parts, then it returns to the left atrium when it gets oxygenated, and after that it enters into the left ventricle, and from this it goes to the whole body with the help of veins or aorta. Okay, students, this is about the heart circulation. So moving on, next question is that the tablet defect arising due to the non-uniformity of color is known as, and the options are blistering, option B is orange peeling, option C is mottling, and option D is capping. So let's the correct answer is mottling. Mottling is the tablet defect arising due to the non-uniformity of color. Okay, and here we have explanation on this, that various types of defects are there. Here blistering, and it occurs due to the rapid evaporation from core, and where orange peel occurs due to the adequate spreading of the coating solution before drying. Students, just try to memorize these defects and their reasons. And where mottling is the non-uniformity of color, in which the question is asked about, and the where capping is removal of crown from main body of tablet. So it's all about the defects and their reasons. Students, try to memorize or write it down in a copy, because many a times the question arises from the tablet portion and from their defects. So moving on, next question. A joint between metacarpal of thumb and carpals is, and options are, option A is pivot joint, option B ball and socket joint, option C is saddle joint, option D is hinge joint, and the correct answers we are having is option C is saddle joint. So let's discuss in explanation that a saddle joint is a synovial joint where one of the bones forming the joint is shaped like a saddle with the other bone and resting on it like a rider on a horse. Saddle joints actually provides the stability to the bones while providing the more flexibility than a hinge or a gliding joint. The bone sitting on the saddle can move in an oval shape relative to the other's bone, similar to the condyloid joints. So let's discuss with the help of diagram that a saddle joint is when one bone that is shaped like a saddle glides along another bone, and this provides a joint with large amount of movement that is relatively stable and safe, and it can move in almost every single direction except for where the concave surface of one joint interacts with the convex surface. It's just having a one exception, so try to memorize it. So it's all about the saddle joint. Next, moving on, next question is example of monocyclic alcohol containing monoterpene is, and here we have options are, option A is geraniol, option B is menthol, option C is nerol, and option D is all. So the correct answer is menthol. Menthol is the example of monocyclic alcohol containing monoterpene unit. So let's discuss in detail about something, that phenyl propane derivatives are safrole, anethole, myristicene, or apiole. And next is bicyclic monoterpene derivative, that is thujone, camphor, pinene, umbelliol, iridoids, and where monoterpene derivatives are menthol, which is asked in the question, limonene, terpenine, and phylendrine and cymene, and acyclic sesquiterpene derivatives are farnesol, nerolidol, and sesquiterpene derivatives are having abscisic acid, cadolin, and curcumin, and bicyclic sesquiterpene derivatives are, here we have caryophyllene, dremanunit, and capsidiol. It's all about the derivatives and their examples. So students, try to write it down in a copy and get revised on time, because very selected question asked from the cognosy portion, and it is very important when we are just talking about the classification of the triterpenoids. So moving on, next question. The first Indian railway line was opened by the British, and here we have options are from Delhi to Bombay, from Bombay to Thane, option C is from Calcutta to Allahabad, and option D is from Bombay to Madras. And here we have option, the correct answer is from Bombay to Thane. The first Indian railway line was opened by the British. So let's discuss about next question, that anti-plaque factor is, option A is riboflavin, option B is niacin, option C is TPP, option D is pantothenic acid, and the correct answer is niacin. Niacin is the anti-plaque factor. So discuss with the help of a table about other factors also, that riboflavin is about lesions and dermatitis, and niacin related to the pellagra, and TPP, that is coenzyme of vitamin B1, is related to the beriberi, and the pantothenic acid, that is B5, is related to the anemia. So the correct answer is niacin. So next question is that the omeprazole acts by, and here we have options are, option A is sodium potassium ATPase, option B is hydrogen potassium ATPase, and option C is sodium potassium 2 chloride, and option D is NaCl, and the correct answer is hydrogen potassium ATPase. So let's discuss about the omeprazole mechanism of action in detail in explanation part. So here we have a mechanism of action of omeprazole, that is that omeprazole is a selective and irreversible proton pump inhibitor, that is PPI. It suppresses stomach acid secretion by specific inhibition of hydrogen potassium ATPase system, and it all occurs in parietal cells, because this enzyme system is regarded as the acid or H+ proton pump within the gastric mucosa. Omeprazole actually inhibits the final step of acid production. So just keep in your mind that omeprazole acts on the last step of the acid production, where omeprazole also inhibits both basal and stimulated acid secretion, and it blocks the last step of an acid secretion, and the drug binds non-competitively, so has a dose dependent effect. So students, just try to keep in your mind that it binds non-competitively. So let's discuss with the help of diagram that actually what's the mechanism of action. That here first just you have to learn is that it's a parietal cell. Okay, it occurs in parietal cells. So here the acid HCl is production is taking place. So how it is possible and how it is going to be regulated, we just take the help of so we have just to take about that histamine. Here histamine is there, it is actually releases by EC cell, ECL cell, that is enterochromaffin cell. So what the work of this histamine, which is regulated from the ECL cell? It actually release histamine which bind on H2 receptor and promote the HCl secretion. Actually it also promote the HCl secretion. So just we are denoting from the plus sign that it promotes the HCl secretion. How it is that it goes to the receptor H2, here we have a receptor, and then with the help of CMP dependent pathway it goes into this and it finally promote the acid HCl secretion. And talking about the gastrin, here we have just gastrin, that G cell releases the gastrin in blood and bind on the gastric receptor which promote the HCl secretion, and it also promote the HCl secretion. So here we are talking about that it promote also the HCl secretion with the help of this pathway, Ca plus 2 plus dependent pathway. So here the HCl secretion takes place. So let's talk about the here then what the actually work of this, that hydrogen potassium ATPase pump, that with the help of this pump there is a production of HCl taking place. So here that it actually exchange H plus ion and it goes H here, then here potassium going outside from this lumen in exchange of H plus, and the HCl coming inside, then H plus and Cl minus combine to form HCl. So it's a all procedure about production of HCl. And what the actually proton pump inhibitors works, it inhibits the exchange of H plus and potassium ion. So if it inhibits this hydrogen potassium ATPase pump, then there is the inhibition of the HCl production. So hence the proton pump mechanism of action is clear, I guess, students. Now the next question is that importance of Krebs cycle is. So let's discuss with the options. Here it is, option A is formation of amino acid, option B is formation of vitamin, option C is increased gluconeogenesis, option D is formation of ATP through oxidative phosphorylation. So the correct answer is option D, that is the importance of Krebs cycle is formation of ATP through oxidative phosphorylation. So let's discuss about the importance of Krebs cycle in our body in detail. So the significance, it is also known as TCA cycle. So it brings about complete and final oxidation of 2-carbon acetyl coenzyme A in the aerobic respiration. It actually occurs inside the mitochondrial matrix, and it is we can also say that it is an amphiphilic cycle, because there in amphiphilic it comprises that catabolism and anabolism takes place in this cycle. Okay, so let's discuss about in detail that it produces simply and harmless end products such as CO2 and H2O. How it is possible that acetyl coenzyme goes into the mitochondria? If the condition is aerobic, actually Krebs cycle actually just work in the aerobic conditions, and the in Krebs cycle the 2 acetyl coenzyme oxidized to form 2 molecules of carbon dioxides. So it is also responsible for the major share of energy releases and supply during aerobic respiration. Out of the total 38 ATP formed during the aerobic respiration of glucose, and Krebs cycle accounts for 24 ATP. Actually Krebs cycle is not going to produce the more ATP in terms of other cycles. So we also discussed some points here, that the TCA cycle produces many important 4-carbon, 5-carbon and 6-carbon organic acid as the intermediate. During this process, students, various types of intermediates form in this cycle, but it not and the intermediates help to formation other end products when the various intermediates formed during this cycle. And as we already told you that it occurs in mitochondrial matrix in the presence of oxygen, and aerobic conditions it takes place only in the aerobes. So it's all about the Krebs cycle. Students, just try to go through the Krebs cycle with the various references books. It is very easy. I hope that this question is clear about the Krebs cycle. So moving on, next question is that 𝙂𝙂𝘖𝑟𝑖𝑛 is A, and the options we are having are: option A is bradykinin inhibitor, option B is cortisone inhibitor, option C is renin inhibitor, and option D is all of the above. So the correct answer is renin inhibitor. 𝙂𝙂𝘋𝑖𝑟𝑖N is a renin inhibitor. So let's discuss in detail that about the 𝙂𝙂𝘋𝑖RIN, what actually 𝙂𝙂𝘋𝑖RIN is. So 𝙂𝙂𝘋𝑖RIN, it is a direct renin inhibitor, which is as a DRI, as the latest class of RAS. What is RAS? Renin angiotensin system. So 𝙂𝙂𝘋𝑖RIN has become available for the treatment of CVS, that is cardiovascular and renal disease, in which ACE inhibitor and angiotensin receptor blocker, that is ARB, angiotensin receptor blocker, and angiotensin converting enzyme inhibitors are currently used. 𝙂𝙂𝘋𝑖RIN, it's all about the 𝙂𝙂𝘋𝑖RIN. Actually, it is a known peptide which binds selectively to the catalytic site of renin and competitively blocks the access of angiotensin to this site. Okay, angiotensin 1 is not produced when and the chain of RAS is interrupted. It's the mechanism of action that 𝙂𝙂𝘋𝑖RIN it competitively blocks the access of angiotensin to this site, where angiotensin 1 is not produced and the chain of renin angiotensin system is interrupted. Here we can explain with the help of diagram that the renin, here it's a renin substrate. When the renin released from the kidney, it converts to the angiotensin 1, and with the help of ACE enzyme, that is angiotensin converting enzyme, and known ACE enzymes such as chymase, it converts into the angiotensin 2, and with the help of angiotensin 2 it goes to the AT1 receptors, and what it leads to the vasoconstriction, aldosterone release leading to H2O and salt retention, and hypertrophic growth, and cardiac remodeling occurs. So it's all about the mechanism of action. So moving on, the next or last question, that is 𝙂𝙂𝘋𝑖RIN equation, which is used for, and the options are: option A is diffusion, option B is potential, option C is diffusion current, and option D is dissolution. So the correct answer is diffusion current. 𝙂𝙂𝘋𝑖RIN equation is used for diffusion current. So let's discuss in detail about this, that 𝙂𝙂𝘋𝑖RIN equation, along with Willis Rodney Whitney, he formulated the 𝙂𝙂𝘋𝑖RIN in the year of 1897, which relates the rate of dissolution of solids to the properties of the solid and the dissolution medium. It is an important equation in pharmaceutical science, and here we have an equation. So it is dW by dt is equal to dA, where dA in bracket CS minus C upon L, where A is the surface area of the solid, C is the concentration of the solid in the bulk dissolution medium, and CS is the concentration of the solid in the diffusion layer of surrounding the solid, and D is the diffusion coefficient, and L is the diffusion layer thickness. So it's all about the 𝙂𝙂𝘋𝑖RIN equation. So the correct answer is diffusion. It's all about